Hello everybody, welcome to Bioscience 360. In this video, we're going to talk about various aspects of amino acids. What amino acids are, what are the different types of amino acids, etc. Our body is made up of many biomolecules like proteins, carbohydrates, lipids, nucleic acids, etc. Now, if we talk about proteins are polymers. Polymer means that it is a chain composed of many repeating subunits. And in case of proteins, these repeating subunits are the amino acids. So what are amino acids? An amino acid is an organic compound characterized by having a carboxyl group, an amino group, and a side chain attached to the central carbon atom. Many amino acids join together to form proteins. So amino acids can be called the building blocks of proteins. There are a total of 20 common amino acids. To make any protein, amino acid residues are covalently linked in a characteristic linear sequence. The word residues here refers to the loss of a water molecule, which takes place when one amino acid is joined to another, making a peptide bond. All 20 amino acids are alpha amino acids, which means that they have a carboxyl group and an amino group bonded to the same carbon atom, and this carbon atom is known as the alpha carbon. These amino acids only differ from each other in their side chains, which is also known as the R group. In all amino acids except glycine, the alpha carbon is bonded to 4 different groups, hence making a chiral center. And when any molecule has a chiral center, the molecule becomes optically active. As we can see in this figure, this carbon is bonded to 4 different groups: one hydrogen, an amino group, a carboxyl group, and the R side chain. Whereas in the case of glycine, we can see that the alpha carbon is bonded to 2 hydrogens, an amino group, and a carboxyl group. Hence, this is not a chiral center, and hence glycine is optically inactive. When we say that any molecule is optically active, we mean that that molecule has the ability to rotate the plane of plane polarized light. There is a tetrahedral arrangement of bonding orbitals around the alpha carbon. There are 4 different groups attached to the alpha carbon, and hence 2 unique spatial arrangements are possible around the alpha carbon, and hence there are 2 possible stereoisomers. These stereoisomers are non-superimposable mirror images of each other, and hence are known as enantiomers. To specify the absolute configuration of the 4 substituents of asymmetric atoms, the DL configuration was developed by Emil Fischer. The DL system is based on the absolute configuration of glyceraldehyde, which is a 3-carbon sugar. All stereoisomers with configuration related to L-glyceraldehyde are designated L, whereas all stereoisomers with configuration related to D-glyceraldehyde are designated D. The amino acid residues in protein molecules are exclusively L stereoisomers. There exists a classification of amino acids based on the properties of the R groups. There are 4 main classes, which are non-polar amino acids, polar amino acids, positively charged amino acids, and negatively charged amino acids. Amino acids such as glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, and phenylalanine exist. All these amino acids have R groups which are non-polar in nature. Glycine has the simplest structure, whereas methionine is one of the 2 sulfur-containing amino acids. Proline has an aliphatic side chain with a distinctive cyclic structure. The secondary amino group of proline makes the amino acid very rigid. The next group includes amino acids having polar R groups. This includes serine, threonine, tyrosine, asparagine, and glutamine. Polarity in serine and threonine is contributed by their hydroxyl groups, and that of asparagine and tyrosine is contributed by the amide groups present in their side chains. The next group contains amino acids having positively charged R groups. This includes lysine, arginine, and histidine. Lysine has a second primary amino group. Arginine has a positively charged guanidinium group, and histidine has an aromatic imidazole group which gives it the positive charge. The next group contains amino acids with negatively charged R groups. This includes aspartic acid and glutamic acid. Both aspartic acid and glutamic acid have an extra carboxyl group which gives them the negative charge. Till now, we have talked about the 20 common amino acids. But apart from these 20 common amino acids, proteins may also contain residues created by some modification of common residues already incorporated into a polypeptide. Such modifications are known as post-synthetic modifications. These uncommon amino acids are equally important for us. Now we are going to talk about 4 non-common amino acids. There are many other non-common amino acids also, but for this video, we are going to restrict ourselves to only these 4 non-common amino acids. The first one, 4-hydroxyproline. This is found in plant cell wall proteins and collagen. The second, 6-N-methyl-lysine. This is a constituent of myosin, which is a contractile protein of the muscle. Next is gamma-carboxyglutamate. This is found in blood clotting protein prothrombin. The last one is pyrrolysine. Pyrrolysine is a special and a rare amino acid as it is not made through post-synthetic modification, but it is introduced during the protein synthesis through an unusual adaptation of the genetic code. Yeah. That's all for this video. I hope after watching this video, you have got a better understanding about amino acids. If you like this video, don't forget to give it a thumbs up and do share it with your friends. Subscribe to my channel for more such videos. Thanks for watching.